Hello everyone. Today we will be talking about the topic of animal improvement via genetic engineering. I'm Afika and together with my group mates, we will be focusing on the ethical and biosafety concern this topic raises. So what is animal improvement? Animal improvement refers to the genetic modification of livestock to enhance productivity traits such as faster growth, increased milk yield, or resistance to diseases. Technologies like CRISPR-Cas9 are often used to introduce or remove genes for these traits. This field plays an important role in global food security and agricultural sustainability. However, we must ask, at what cost? There's a growing ethical tension between scientific progress and what's morally acceptable. Just because we can genetically modify animals does not mean we always should. Some key ethical issues include animal suffering, where animals may experience pain, miscarriages, or physical deformities due to gene editing. Others worry about the loss of animal integrity, as animals are treated more like biological tools than sentient beings. There's also the potential for long term ecological disruption, especially if genetically altered traits unintentionally spread into the wild. Beyond ethics, there is also the biosafety concern that we must take seriously. Let's start with animal welfare. Studies have shown that cloning and genetic modification often have very high embryo loss rates. Those that survive may suffer from malformation or shorter lifespan, and in some cases, the genetic changes affect behavior and physiology. Now, looking at biosafety risks, there's also a chance for horizontal gene transfer, where modified genes might escape into the wild population. Another issue is the use of antibiotic resistant marker genes, which can potentially contribute to the broader crisis of antibiotic resistance. And finally, these genetic changes could affect the human food chain, for example, by introducing new allergens. A real world case is the EnviroPig, a genetically modified pig developed to digest phosphorus more efficiently. While scientifically successful, the project was shut down due to public and ethical backlash.
Next, we'll go into how the three R's replacement, reduction, and refinement guide more ethical approaches to genetic improvement in animals. Let's start with the first R, replacement. This encourages the use of alternatives instead of live animals, especially in early research stages. For example, in silico models, advanced computer simulations help predict how gene edits might affect traits like growth or disease resistance. Another tool is organoids, like grown 3D tissues that mimic real organs. This allows scientists to study gene expression and disease without using full animals. Now, the second R, reduction. This focuses on using fewer animals while still getting reliable data. With genomic selection, researchers can identify the best candidates for testing before trials begin. And shared genomic databases help labs avoid repeating studies by using existing data. Finally, refinement. When animals are involved, this ensures better welfare. Non-invasive genotyping using hair, saliva, or feces avoid painful procedures. And better handling practices, pain relief, and shorter trial durations reduce stress and suffering.
Now, let's talk about the new technologies which allow us to continue studying or improving animals genetically, but without relying so heavily on live animals. These technologies follow the three R's, which are safer, more ethical, and more accurate. So to start, with in vitro fertilization combined with embryo screening, we can now screen embryos early and select only the viable ones instead of implanting genetically modified embryos and waiting for full development. This reduces the need to develop and sacrifice entire animals just for testing purposes, directly aligning with the replacement principle in the 3 Rs. Next, by pairing CRISPR gene editing with AI, we can predict off-target mutations before experiments even begins. This means we can prevent failed edits, avoiding suffering or unnecessary animal use. It also boosts precision, reduces error rates, and supports both refinement and reduction in animal research. Digital twins are virtual models that replicate the full physiology, metabolism, and genetics of a real animal. These simulations allow researchers to test how genetic modifications might affect an animal without ever touching a real one. This kind of in silico modeling represents a major step forward in replacement strategies, especially for early stage experiments. Last but not least, induced pluripotent stem cells or iPSCs can be derived from livestock cells and grown in the lab. These cells mimic real tissues and can be used to study genetic effects or responses to treatments. Using iPSC models reduces the need to breed or modify live animals for early testing, fulfilling both replacement and refinement principles. To conclude, applying the three R's is essential for ensuring scientific accuracy, public trust, and regulatory compliance. So, call to action. We must integrate the three R's into all animal research policies. We must invest in alternative technologies that reduce or eliminate animal use. Last but not least, we must prioritize animal dignity alongside human benefit. So, that's all from us. Thank you.